Hi all, so prokaryotes, let's talk about what we're going to be looking at today. So we're going to look at what are prokaryotes, prokaryotic diversity, bacterial cell division and communication, mechanisms of bacterial disease and how antibiotics work, which is our main way of treating bacterial infections. So bacteria have been around for a long time. We think they're one of the earliest forms of life. And they've been incredibly successful. They're really important in an ecological sense, both in the carbon cycle and the nitrogen cycle. In the carbon cycle, acting as decomposers, breaking down dead organic matter, that is detritus, and returning both the mineral ions to the soil, but then the carbon dioxide back up to the atmosphere. Remember, an absence of decomposers is going to cause an accumulation of dead organic matter, which over a few million years could result in fossil fuels. That's where our fossil fuels have come from. But remember, bacteria, along with fungi, are primary decomposers, breaking down that dead organic matter and releasing the carbon dioxide back into the atmosphere. In the nitrogen cycle, bacteria have a few important jobs. Arguably the most important of these is nitrogen fixation. This is taking inert, effectively useless atmospheric nitrogen and converting it into nitrates. Nitrates are the form of nitrogen which plants can take up and convert into amino acids for building proteins, right? Nitrogen is part of the amino group. They can convert this nitrogen into nitrogenous bases, which of course are used in nucleic acids too. And we get our nitrogen from plants. So it needs to be in this reactive form. Bacteria also play an important role in then releasing that nitrogen, which is normally bound in the form of ammonia or urea, back and converting it back into nitrates. These are called nitrifying bacteria. There are some bad, effectively, bacteria in this cycle, and those are denitrifying bacteria. They take nitrates, which remember are really important for plant growth, and convert it back into atmospheric nitrogen. This tends to happen in waterlogged soils. Converts it back into atmospheric nitrogen, effectively making that soil a lot less fertile. So this is really bad news for farmers and why drainage in farming is so important. Just a side note, lightning can also convert inert, that's unreactive atmospheric nitrogen, into nitrates. So lightning is good for farmers. Bacteria, of course, though, are very important to us as well. And directly to us. The images on this slide come from a TED Talk. I really recommend watching this on how bacteria communicate. And it's by Bonnie Basler. And one of the things that she talks about is just how numerous bacteria are on us, right? You've got about 1 trillion bacteria in your body. My bad. You've got about 1 trillion human cells in your body. So when you think about yourself, you think of, I'm human. But actually, if we go by cell count, there's about 10 trillion bacterial cells. So way more cells that are on your body and in your body are bacterial than they are human. Now, you may think you've got the beat of them there when it comes to genetics. You've got about 30,000 genes in each one of your cells. But if we look at the total amount of DNA present in and on your body, there's actually about 100 times more bacterial DNA. So both in terms of a raw cell count and in terms of the amount of DNA present on you, actually you've got a lot more bacterial DNA than human DNA, which is a strange thought. And obviously, we need to slightly maybe reconsider our own personal identity. Let's talk about bacterial structure now. So bacteria are prokaryotic. Prokaryotic means not having a nucleus. So the DNA is loose within the cytosol. The chromosome is in a loop form, unlike the straight forms of our chromosomes. And it's found in the middle, which we call the nucleoid region. But it's important to remember that there is no nuclear membrane around this chromosome. So it's the single loop of DNA. It's a big loop of DNA. There is other DNA present in the cytosol too, and this is in the form of plasmids, these small loops of DNA. And these are like subsidiary programs for the cell. If you think of the chromosome as being the core operating system, the plasmids are like apps, effectively. They enable to do the bacteria to have extra functions, to grow new body parts, and that might be something such as a flagellum. You can see the flagellum here coming off the back. The flagellum is a whip-like structure that enables a bacteria to be propelled along. Now, the flagellum is the kind of thing that might be encoded for by a plasmid. As well as that, on the exterior surface, we have pili that extend through this capsule or slime layer. Pili are used for bacterial conjugation, and we'll come to that in a bit. The capsule or slime layer, again, it's one of these things that can be coded for by a plasmid, provides the bacteria with protection. And then underneath it we have a cell wall, not like a plant cell wall which is made of cellulose. This cell wall is made of a carbohydrate called peptidoglycan. We also have a plasma membrane to control, obviously, what enters and exits the cell. Inside the cytoplasm, along with the plasmids and the chromosomes, you'll also find ribosomes. These ribosomes tend to be a little bit smaller than eukaryotic ribosomes. Yeah, they're very important. Remember, this all relates to the theory of endosymbiosis. Remember, mitochondria and chloroplasts in our own cells have their own DNA, which is in a loop, like bacterial DNA. They have their own ribosomes, which are smaller, like bacterial ribosomes, and they have a double membrane, suggesting that at some point in our evolutionary past, chloroplasts and bacteria were once independent prokaryotic organisms that were absorbed, taken in by a larger eukaryote-like organism. And this happened sometime in our past, we believe. So there's two main groups of prokaryotic organisms. There are the eubacteria, and these are your kind of classic bacteria that you think of when you think of bacteria normally. Most of these ones are ones that you're familiar with. They grow in your gut. They may give you diseases, etc. And then there's the other group of bacteria, or prokaryotes, called the Archaebacteria, or sometimes just the Archaea. And the Archaea used to be just clumped together with the bacteria in terms of human classification. But now we're starting to understand that actually the Archaea are quite a distinct group. And the Archaea tend to be extremophiles. We find them if you go to Yellowstone National Park, for example, in the incredibly hot springs there. The bacteria, the prokaryotes that are growing are Archaebacteria. And we find them in extremely hot environments, extremely sulfurous environments, perhaps incredibly salty environments, or high salinity environments. So these tend to be quite extreme organisms, and it's actually where we get some of our key tools. For example, in PCR, we take some of the DNA polymerases from these extreme thermophiles because we can then have thermostable molecules. So these two distinct groups, we believe, split off relatively early in evolutionary history, and there is some evidence from ribosome structure and some DNA evidence that eukaryotic cells are actually more closely related to the Archaea than they are to the eubacteria, the regular bacteria. Bacteria can be divided and classified in part by their cell structure. We have the gram-negative and the gram-positive bacterial cells. And this is to do with their cell wall structure. The gram-positive ones have a thick peptidoglycan wall outside of their cell membrane. This means that when we apply a particular stain to them, a gram stain to them, they show up as darker cells. These ones tend to be easier to treat with antibiotics because the cell wall is exposed. It's a structure that we can attack with antibiotics. Gram-negative bacteria, on the other hand, have this outer membrane layer with the peptidoglycan underneath and then a secondary cell membrane underneath that. That means that the peptidoglycan layer is not as close to the outside, so when we stain it, they tend to have a fainter stain, so they don't show up as strongly. And it means that generally these bacteria are harder to kill with antibiotics. So some of the particularly nasty diseases tend to be gram-negative ones. Bacteria, as you can imagine, being incredibly small and are very difficult to classify. They're difficult to classify. We're getting better at it as we look at their genetics, but they're able to freely exchange bacteria, and where the boundary is between one species and another can be pretty difficult to draw the line. One of the ways that we use to classify them is by their shape. We have the cocci, which are these round ones. They can form clumps. Okay, so for example, Staphylococcus aureus forms these flat sheets like so. Others will form big blocks of them. Streptococcus is this long strand of the cocci. There's bacilli, which is your classic rod-shaped bacteria. The long rod shapes, there's a few examples on there, such as Salmonella. And then we have a few other shapes, the Vibrios, the Spirellae, which are like spiral-shaped. So those are the three main types of the Spirellae, the bacilli and the cocci. Round, oblong, oval shapes, and then the spiral shapes. When bacterial cells divide, they need to do DNA replication beforehand, just like our cells do. But their replication forks obviously will go around that loop and meet at the termination of replication at the other side. Similar for plasmids too, so dual replication forks in DNA. Bacterial cell division is a relatively simple process, therefore, because of course there's no nucleus to break down. So the cell literally will just bud off and split into two after it's copied its central chromosome. This is called binary fission, and while it might not seem the most exciting thing, remember, it's still better than Twilight. So a little video for you to watch here showing some bacterial cell division. Bacteria can divide very
reproduce
fast.
very simply and rapidly by
They can divide approximately every 20 minutes under ideal conditions, and so by binary fission can, under ideal conditions, create many billions of copies very, very quickly if they have enough room and enough food to grow. Bacterial conjugations, this is an important topic. This is effectively sometimes known as pseudosex. Bacteria are able to exchange plasmids with one another. So these kind of apps, these extra pieces of information that are stored on these small loops of DNA, can be copied and swapped from one bacteria to another. So you can see on the left we have a diagrammatic representation of how this happens. The DNA strands separate, and one of the two strands, just one of them, is passed to the other bacteria. While that is happening, both of those strands then are being copied, and so both bacteria end up with a copy of the original plasmid. Here's a photograph on the right showing this actually happening between two bacteria. This obviously enables them to pass those extra attributes, such as a capsule or slime layer, a flagella, and share that with their neighbors. This means that bacterial evolution can happen very, very quickly, even faster than it would otherwise, because they can quickly spread genes throughout a population. Speaking of populations and how bacteria relate to one another and communicate, one of the things that have been recently discovered is that bacteria behave differently given their population density. Effectively, what happens is they send out signals into their surrounding tissue, such as this HSL signal, that basically kind of says, I'm here. Now, if there's not many of them around, then the concentration of HSL will stay low, and the bacteria will remain in a planktonic state. They will behave in a particular way. But if there is a high concentration of these signals, these chemical signals, then the bacteria switch their behavior. In other words, they are able to sense a quorum, a group of them together, a critical number, and when they reach that, their behavior changes. So an example of this is the plaque that forms on your teeth when a lot of bacteria in the same area They lay down this kind of cement that helps to hold them in place, and they don't behave like that when they're on their own in suspension, but when they're together on the surface of your teeth, they create this biofilm, as it's known. So speaking of bacteria harming our body, there's a lot of different types of bacterial disease, and I just want to run through a few of those. You may have heard of strep throat. Streptococcus bacteria they're the most common bacterial illnesses. Cholera, caused by Vibrio cholerae. Cholera is a particularly interesting one. It uses a two-part toxin to induce a new cell signal pathway to exploit a cell signaling pathway in your intestinal cells, which causes water to flood into the lumen of your intestine, giving you diarrhea and enabling the bacteria to move out of your body to a new host. It's its way of spreading, a particularly interesting one. So this is spread by the fecal or oral route, which is basically feces going into your mouth somehow. Now, not normally directly, but contaminated drinking water, contaminated food, tends to happen in areas where there's large disasters, such as the earthquake in Haiti. There was an outbreak of cholera after that due to contaminated drinking water. Salmonella, which causes things such as food poisoning. Staphylococcus aureus. So Staphylococcus has become one of the most notorious antibiotic resistant strains of bacteria, MRSA, which is common in hospitals. Enterobacter cochleae. So this is urinary tract infections. We get bacteria that cause meningitis. Haemophilus influenza doesn't actually cause the flu, causes meningitis in children in particular. We have bacteria cause the Bubonic Plague, the Black Death as it was called, and that's actually that bacteria is still found in squirrels and interestingly in rabbit populations in the US still persists there. So if there's a rabbit in your yard, it may well be carrying the Bubonic Plague. We have Helicobacteria, which are cause more intestinal problems. We've got spirochetes, so these are sexually transmitted bacteria. So syphilis is one of these, and then there's another sexually transmitted bacteria, the ones that cause gonorrhea, for example, are transmitted that way. So the way that these bacteria cause disease in your body is, first of all, they need to latch on to you. So they use these thread-like extensions called fimbriae, and they attach it to mucous membranes. The mucus normally manages to keep the bacteria off, but using these threads, they can attach themselves. And then they're going to release digestive enzymes to break down the connective tissue around, allowing the spread of the infection and allowing the bacteria to feed off your body's resources. When you have a blood clot, your body releases fibrin. These are fibrous proteins that their intention is to not only reduce the flow of blood out of the body, but also to trap the bacteria in place. But bacteria have a countermeasure to this. They can release enzymes that break down the fibrin. And so if you think of a cut that has got infected, it doesn't tend to form a hard scab, and that's partly because of these enzymes that are being released. White blood cells have come in different forms. Phagocytes are the ones that consume bacteria, and phagocytes patrol our body looking for bacteria, looking for foreign bodies to consume. Again, bacteria, though, do have defense mechanisms against phagocytes, such as toxins that will destroy white blood cells. So this is an ongoing battle. It is important to remember, though, at this point, that there is this huge population of bacteria on our bodies that actually is incredibly helpful to us. That TED talk I mentioned earlier goes into a bit more detail on some of them. But it's really amazing how they help us, both in terms of supplying us with vitamins, or vitamins as you call them. We have symbiotic relationships, lots of these, and so mutualistic symbiotic relationships. So don't put all bacteria into the bad camp. Okay, they're not all bad. So when we do have bad bacteria that cause disease, how do we treat them? We use antibiotics if it's internal, and these are difficult drugs to develop because they need to target the bacterial cells but not our own body cells. So antibiotics exploit differences between prokaryotic and eukaryotic cells, such as cell wall synthesis. Penicillin, the first first antibiotic that was used on a wide scale, attacks the cell walls of prokaryotic cells. But there are others, and you can see them here, that attack DNA replication, transcription, the membrane, and processes of translation. All processes that the bacteria will need to be successful. It's very difficult to develop new antibiotics, though, and part of the problem with this is that you don't want to be destroying your own body cells, but two, bacteria evolve very quickly. And we've seen a large increase in the amount of antibiotic resistance, often held on these plasmids. So, for example, the plasmid might code for an efflux pump that pumps the antibiotic back out of the cell. It may code for an enzyme that alters the antibiotic or degrades the antibiotic, and so or creates a protective capsule that prevents the antibiotic reaching the bacteria. And the thing is that as we use antibiotics, in particular this overuse in agriculture, as we have pigs and cows being given antibiotics to decrease the disease amongst them, we're using the antibiotics so much that it's actually creating natural selection. Those bacteria that are more resistant are more likely to survive. So, for example, in this case, there's an antibiotic with H. pylori that attacks and causes this particular enzyme in the bacterium to induce bacterial cell death. But the mutant form that doesn't have this enzyme survives, and so in the next generation of bacteria, the antibiotic resistance is much, much more common. And so we're seeing a rise in these problems and diseases such as MRSA that we just cannot treat, and that's a big problem. So to finish with, prokaryotes, evolving quickly, a huge part of who we are, and make sure you know all the different parts. Make sure you understand bacterial conjugation and the interaction between antibiotics and bacterial resistance.